Drosophila is another very popular model organism. I made the point in my first lecture that many of the advances in genetics, starting just after the turn of the 20th century, were made in Drosophila. A lot of tools were developed in Drosophila, and I'm going to talk to you about some of the current tools that people are using in Drosophila beyond just the targetable nucleases. The question is, why Drosophila? The answers have been scrambled. One answer is, as I recall, that a lot of gene discovery has been done in Drosophila. So Notch, discovered in Drosophila. Toll-like receptors, discovered in Drosophila. Hedgehog, another important pathway, discovered in Drosophila. And so Drosophila has been an important place for gene discovery. It's also been an important place for working out gene function. So the homeo box genes were discovered and their function was worked out in Drosophila. The polycomb complex that I listed here, the broad repression complex in genomes throughout the biological world, that was discovered and worked out in Drosophila. Many human diseases have been modeled in Drosophila, and I'm going to show you, I hope, some examples of that, depending how things go, including Huntington's disease, some aspects of addiction, neuromuscular diseases. There's also been quite a bit of technology development in Drosophila using transposons and recombinases and the FLIP-FRT system. These things have been used in Drosophila, and the zinc finger nuclease is actually the first organism in which the zinc finger nucleases were ever used, even before they were used in cultured mammalian cells, was the fruit fly. And one of the reasons for all of these nice things happening so comfortably in Drosophila is that genetic analysis is very easy. And this is a poster that we actually have hanging in my lab, probably hanging in a lot of Drosophila labs. It's a gallery of mutant phenotypes in Drosophila, and there's no reason to look at all of them, but I had talked before about the wild type I having this nice bright red color, the white mutant that was identified in the Morgan lab well over 100 years ago, the rosy eye color that we use commonly in my lab. And then there are some cuticle colorations. This is the wild type cuticle, yellow mutant, ebony mutant. There are some bristle phenotypes that I'm sure you can't see. This one is stubble. I think this one is singed. And the genes that are responsible for these phenotypes, first of all, are single genes. Some are recessive and some are dominant, or there are mutations that are recessive or dominant in these genes that are inherited in a Mendelian fashion. So it's been quite easy to use the simple genetic markers throughout the Drosophila genome to help with genetic analysis. And these are just clearer illustrations of some of these mutant phenotypes. This is the white mutation. One we use quite a bit is a dominant allele called curly that makes the wings curl up like this. And there are chromosomes that are marked with curly, and you can trace the inheritance of that particular chromosome through infinite lineages just by the fact that the wings have this odd appearance, and it's a single allele. The homeo genes, these homeobox genes, were discovered as gene complexes that controlled these homeotic transformations. They were studied particularly by Ed Lewis at Caltech. And this is an example called antennapedia. So instead of, in this mutant, instead of just losing the ability to grow antennae, which would normally grow out here, this mutant now has legs growing out from the antenna buds. And so there's been this sort of whole organ transformation that has occurred, and it's as a result of regulation by these high-level regulatory genes that have homeoboxes in them. And the homeobox comes from homeotic transformation. This is an example of using Drosophila to study human disease. Many of you are probably aware of these triplet repeat expansion diseases where triplet with, not always, but often within coding sequence, a multiplicity of triplets will be expanded, and as that cluster gets larger, there gets to be a disease of some kind. And this is looking at one of these triplet expansion examples where the triplet that's expanded is a glutamine codon, and so you get what are called the poly-Q segments in the protein, and the protein that's being used here comes from a spinal cerebellar ataxia type 3 and driving that poly-Q protein with an I-specific promoter. And what happens with these proteins is they aggregate and they gum up the proteasome and they do bad things to cells, and they even do bad things to Drosophila eye cells. So this is a normal eye. This one's expressing the poly-Q protein. And, the pigmentation is gone. The organization of the segments in the eye, the ommatidia, has been lost. And one of the reasons for setting up something like this is so you can see if you can fix it. And one of the things that makes a positive difference here is overexpressing a chaperone, in this case HSP70, a heat shock protein. And Nancy Bonini has been a leader in this area of using the Drosophila eye as a place to study disease mutant phenotypes. So this, having ommatidia in your eye go away isn't something that happens to humans who have this phenotype, but at least we can study how aspects of how the phenotype is caused. So now I'm going to talk about some of the tools that have been developed in Drosophila, some of which have been exported to other systems, but others have not. But it gives you an idea again of how Drosophila can be used in very intentional ways to study things. This is the GAL4 UAS system that was developed quite a long time ago by Andrea Brand, working in Norbert Perimon's lab at Harvard. So the basics of the system are that you take components from yeast, the GAL4 gene activating protein, and its binding site, which is called an upstream activating sequence, or UAS. And you endow whatever gene you're hoping to regulate the expression of with upstream activating sequences, usually multiples of it. And at the same time, you can put in a transgene for GFP that also is being regulated by the UASs, and then express GAL4. And when GAL4 is expressed, it'll bind to these binding sites and turn on both gene X and GFP. The advantage to doing this coordinately is that you can see the cells that are expressing GFP, and those will also be the cells that are expressing protein X. So that sounds like kind of a boring thing to do, except for the fact that this GAL4 protein has been put into the Drosophila genome driven by a lot of different promoters. And some are ubiquitous promoters, but some are tissue-specific or development timing-specific promoters, so that you only get the driven expression of these genes in particular tissues at particular times in development. And there's a further layer of control here that is provided by another yeast protein. It's called R here. It's actually the product of the GAL80 gene. So GAL80 normally interferes with gene activation by GAL4, but there's a temperature-sensitive version of GAL80 that will interfere with gene activation until you go to a higher temperature. And the temperature differential here is only 4 degrees, from 25 up to 29. You can get temporal control of these genes within the laboratory by using a heat shock to turn a gene on. So you have exquisite control over when and where a transgene is active within the flies. This is an example where that same approach has been used to turn on interfering RNAs, these short hairpin RNAs. So you can put the UAS in front of these RNAi expression constructs, and then just turn on RNA interference in a tissue and timing specific fashion. And this gallery, this is from a review. This gallery shows patterning in the Drosophila embryo in the wild type, and then after turning on with an early development specific promoter, RNAis for different genes. And this is Nanos, Krupel, Fushitaratsu, decapentaplegic, these different genes that are involved in the development and the patterning of the Drosophila embryo. So, trying to remember the year, Alan Spradling and Jerry Rubin in the 1980s sometime developed a transgenesis procedure for Drosophila. And the way this works is that they take a natural Drosophila transposon and use its ends to incorporate any piece of DNA, any gene you want. And the transposon that was initially used was called the P element. People have devised other transposons for Drosophila now. So you can pack a P element with any collection of sequences you want, and then just having the ends of the P element surrounding that collection of sequences, if you express the P transposase, at the same time you put in this DNA, typically as a plasmid, that hybrid transposon will go and it'll jump into various places in the genome. That was fine for making stably integrated transgenes, but depending where the transgene went, its expression level could be high, intermediate, or low, because particularly in Drosophila, the chromatin context has a big effect on gene expression, and this is called position effect variegation. So where your gene goes, where it is located in the genome, determines how much it's expressed. So that was an issue for many years for transgene insertion. You're trying to put in a gene that's going to complement a mutation, and it may complement it well or poorly depending on where it went. But people took advantage of this in more recent years by putting in landing sites for an integrase that comes from a phage called Phi-C31. So this at-P site is a target site that the Phi-C31 integrase will recombine with another sequence called at-B. So this is like the lambda integration system. There's a bacterial attachment site and a phage attachment site that are different from each other, but the integrase recognizes the two and recombines them. So what people have done is to put this landing site in different places around the genome, not in the same fly. Each different strain of flies has a single landing site. And then what you can do is you can put your gene into a multiple cloning site in this vector and integrate it specifically at one of these locations. And every time you make a modification here, either with a different gene or a different version of the same gene, it'll go into the same landing site and have the same chromatin context, and within some range have the same level of expression. So it's a way to overcome this position effect variegation. And the vectors here have gotten more and more complicated, but they're set up often so that you can excise all the stuff you don't care about in the neighborhood after you've gotten your transgene into this very specific location in the genome. So those are some of the tools. Now in tools we're most interested in. I'll go through this pretty quickly. The very first use of engineered zinc finger nucleases in living cells or organisms was this one. This hasn't happened to have happened in my lab. Marina Bibikova and John Troutman and I collaborated. Well, we asked for help from Kent Golick, who's a real Drosophila geneticist, and we built zinc finger combinations and built them into the zinc finger nuclease configuration for a target within the yellow gene of Drosophila. So I mentioned before, yellow controls the color of the cuticle and bristles on the fly. And we expressed these two zinc finger nucleases in Drosophila. And at that time, we were using sort of a complicated system where everything had to be integrated into the genome before it could be turned on. And I won't belabor this, but we set it up so that when the nucleases were expressed and cut the target, that a donor DNA, which was also integrated in the genome, was excised by FLIP and linearized by ISCE1, so it became a linear donor. And I think I mentioned on Monday that there's a single copy of this donor in every cell. And the fact that we got homologous recombinants out of this at a reasonable level is completely remarkable to me. One copy of the donor, one copy of the target in males, and still we were getting good levels of recombination. And we could see the effects of mutagenesis at the yellow locus, even in the initial generation where the heat shock induction was done, because there are these patches in a normally completely darkly pigmented cuticle at the end of the male abdomen here. We could see these patches of light tissue, lightly colored tissue, which are characteristic of yellow mutations. I mentioned on Monday that our workhorse in Drosophila has been the rosy locus, where we made a different pair of ZFNs and a donor DNA. And shortly after we began working with Rosie, we also had a different method for introducing ZFNs or talons or Cas9, and that's direct injection into embryos. So in this case, you can take the injected flies once they grow up from those embryos, cross them to a reporter strain of some kind, and evaluate germline mutagenesis in this next generation. And the generation time of Drosophila is about 12 days, something like that. So you can get through experiments pretty quickly, not as quickly as in the worm, but pretty quickly. And so I think I showed this before. We expect to get mutations by non-homologous end joining and by homologous recombination with the donor DNA. This is just illustrating the cross that's done in diagrammatic fashion. We've crossed these candidate flies to genuine rosy mutants, that actually usually a deletion, non-revertible mutation, and score the rosy phenotype in the next generation. We can score for homologous repair by the integration of that restriction enzyme site, rosy versus wild type, easy to screen. And with the injection procedure, these are injections done in my lab. 99 flies grew up from those injections. 41 of them gave new mutants, and we got lots and lots of new mutants. And when we sent our materials off to a company in Cambridge, Massachusetts called Genetic Services, they were even more effective at generating new mutants by this injection procedure. When you don't have a visible phenotype to score, I mean, we were using Rosie because we could see it, you know, we could do it over and over again. We probably made more Rosie mutants than all the rest of Drosophila geneticists anywhere in the world. But if you don't have a phenotype, the frequency of mutagenesis is high enough that you can do a straightforward molecular screen to find them. And the way we do it takes advantage of tools for fly genetics. So this is the injected fly or group of flies. And once they get to be adults and we cross them, we can evaluate them to see whether there are mutations anywhere in the soma of these flies, which is an indication that something good happened during embryo injection early on. But we cross them to flies that on their 2 chromosomes carry a dominant marker that we can always see and a deletion. It's often called a deficiency. That's why it's deficiency here. It should be, I should have put the delta in there too. I did that this morning. So if you generate knockout mutations in your target and they're in an essential gene, you won't get very many of these guys because they have a deficiency, a deletion that covers the target locus. And if you made a lethal mutation, you won't get these guys. But you can use them, again, to do molecular analysis to see whether there are any mutations that are being transmitted from these guys. Here you're only looking at somatic mutagenesis. Now you're looking at something that had to be transmitted in the germline. But you can also take these candidate chromosomes, which have been isolated over this dominant marker, and cross again. And now, from individual flies, you get to score them over the deletion to see whether they're viable or not, and also to do a more detailed analysis of the mutation. And HERMA, you remember, is this analysis where you do high-resolution melting after PCR of the locus, reannealing to get heteroduplexes, and then looking at just at a melting curve. And the melting curve isn't telling you what the mutations are. It's just saying whether mutations are there or not. And these are some data from Kelly Bumer in my lab, just showing that evaluating individual flies from the initial injection and comparing them to wild type, we see a lot of these individual flies carry mutations that are detectable by HERMA. And then when you evaluate individual offspring of those flies, now you get more orderly patterns because each of those is only carrying a single mutation. So, but still you can use this high resolution melting as a way to screen to see who are mutants and who are not. This is a set of experiments we did to test the talons in Drosophila. And the bottom line here is that of the 17 different talon pairs we tested by doing messenger RNA injection into the Drosophila embryos, 15 of them worked well enough to give usable levels of mutation. And so this includes rosy and yellow, but PSF2 and SLID5 were genes that we targeted for Mike Botchin's lab here in Berkeley. A couple of these down here are genes we targeted for Scott Hawley, who's in Kansas City. So, you know, pretty much any target we decided to hit with talons, we had a very high success rate for, better than we had with the ZFNs. So this is a reflection of the advancement of the technology from ZFNs to talons. We were far from the first to use the CRISPR-Cas9 system in Drosophila, so I'm going to tell you mostly about things that have been done in other people's labs employing the CRISPR system. So this is, you know, the Doudna-Charpentier paper demonstrating that RNA-guided Cas9 is a sequence-specific DNA cleavage tool, and sort of opening the door to what people have been doing ever since. That was published in the summer of 2012. By the end of 2013, there were lots and lots and lots of papers using the CRISPR system in a lot of different organisms and cell types, and there were a whole bunch of papers in 2013, starting, I don't know, in June or something like that, employing the system in Drosophila, and this is one of the earliest papers. So what they're doing here is they're just showing that they can hit the yellow gene with guide RNAs that are targeted here. That's very close to where we were targeting ZFNs, and also the white gene, the gene that is responsible for eye color. one of the genes responsible for eye color. And using these different guide RNAs, even in the injected generation like we were seeing, they could see these somatic mosaics where there are patches of mutant tissue surrounded by wild type tissue. This is yellow. And it's even more obvious with white, where you have this somatic mosaic of pigmentation in the eye. And these are both genes that are whose products function cell autonomously. So every cell that gets a mutation is going to express the absence of the phenotype, and there are other genes in many organisms that don't function cell autonomously but would affect the entire organism. So another paper published 2 years ago in the summer set up another way to express Cas9 and the guide RNA in flies. What they did was they made a transgene for Drosophila that has Cas9 driven by this nanos promoter, which is a germline specific promoter expressed in both the male and the female germline. So you make animals that are carrying this at one of those landing sites that I was talking about earlier, and then you make another line of flies that have the guide RNA you're interested in driven by an RNA polymerase III promoter, the U6 promoter. And then when you cross these flies, all of their offspring get both those genes, and mutations will be induced to the extent that that's a good guide RNA and that's a healthy Cas9 protein, and you can just screen these. And this is an example of using a situation like this where you actually have two guides encoded in the genome of one of the parental flies, and you get deletions induced by cleavage at both of those sites. And this is just the PCR products indicating how much DNA has been deleted, and I'm sure they sequenced those. So this is looking at numerology for targeting different sites within the white gene. So these are the guide RNAs. That's not a white gene guide RNA. And the first column just says how many of the parents that were injected with that particular guide RNA, or, sorry, this is a cross. So how many of the parents that were involved in a cross with this particular guide RNA gave new white mutants? And the answer in all cases was they all gave new white mutants. How many of the progeny of those flies were new white mutants? And the answer is that some of them worked pretty well, and some of them worked like gangbusters. Here, there are only 5 flies out of nearly 1,000 that weren't new white mutants. And this is in the male germline. In the female germline, things were always a little lower. Any idea why? What could have caused that? Huh? It's on the X chromosome. The target is on the X chromosome. So to get, and the mutations are recessive. So to get the white mutant phenotype, you have to knock out 2 alleles in females, but only 1 in males. Sorry, that was a genetics question. Okay. So these folks set up a system that makes use of this GAL-UAS procedure, so that you can induce the expression of Cas9 not throughout the fly that you're working with, but in specific tissues or at specific times in development. So this is just saying, okay, we've got a GAL4 that's under the control of a particular enhancer and promoter. We cross that with a fly that's carrying a guide RNA driven by a U6 or a different RNA polymerase III promoter, and Cas9 under UAS control. And so this is looking at examples of this, driving guide RNA for the yellow gene with a ubiquitous promoter, and you get a range of mutations throughout the fly, but a lot of them are completely mutant because of the fact that it's a ubiquitous promoter. So to target Notch with a guide RNA, they used a promoter that was specific for the wing margin. So this illustrates a nice feature of this Gal4 UAS system. Notch is important for early development in Drosophila. So if you completely knock out Notch, you don't get adult flies. But to see the effect of Notch in a specific tissue, you can drive Cas9 with a tissue-specific promoter and just see what the effect is on the wing. You can also see from this slide why this gene was called Notch, right? Because even in the original spontaneous mutant, the wings were notched like that. And now people identify it as a sort of a neural development specific signaling factor, but it was originally discovered in Drosophila. And people have used this procedure. This is called Cas9 mediated conditional mutagenesis. Sorry, I forgot what the other C was for. To drive the mutagenesis of genes important for germline development drive the expression of Cas9 with a germline specific promoter, again, so you can isolate the effect on the germline from effect in other tissues. And this is showing actually driving Cas9 with a nanos promoter and then targeting the nanos gene. And this is another germline specific gene, and this is wild type cases where there was no apparent mutagenic effect and then different severity of mutation, and they're showing you the scoring here. And I think this was done with that heat-inducible Gal4, so you get more mutagenesis, more severe mutagenesis at 28 degrees than at 25. People have used the Cas9 RNP approach in Drosophila as well. This is a paper from Jinsoo Kim's lab. Here they're targeting the ebony gene. And in ebony mutants, the cuticle, this is the thoracic cuticle, is darkly pigmented compared to the wild type. And so they designed a guide RNA to a critical sequence within the ebony gene, and were able to generate ebony mutants. The problem with this was that the efficiency was actually quite low compared to either injection of messenger RNA plus guide RNA into the embryo, or these methods where you have things in the genome and you're doing a cross. One hybrid procedure that people use a lot that I didn't describe is to use Cas9 in the genome under some, usually under a germline promoter or a ubiquitous promoter, and then inject a synthetic guide RNA, and that also works quite well. Okay. This I thought was an interesting use of the Cas9 mutagenesis in flies, and it illustrates how finely you can direct your questions. Barbara did a nice job of showing some of the things they did at very, very specific sites in worms. Here's another example, and this is one that has also been used, at least in a few instances, in mammalian cells. And the issue here is I've got a pair of microRNA and target gene, and I only know that they're a pair because I know the microRNA is made and I know the gene is transcribed, and it looks like a nice seed sequence for binding of the microRNA to this gene, but I really want to know whether the microRNA controls expression of the protein made by this messenger RNA or not. So these folks targeted Cas9. This is the PAM here. Then the cut will occur there. Targeted Cas9 very close to this seed sequence in the protein coding gene. So they're not actually, in this case, they're not actually hitting the microRNA gene, they're hitting the target. And so they do the embryo injection, they do HERMA, they get out mutant flies, they sequence the mutants, and they get quite a number that will have deleted this seed sequence, and some that will have made other sequence changes in the vicinity. The microRNA is called Bantam. I don't remember what ENA stands for, but that's the target gene. And then they look to see, first of all, what's the region of expression of the microRNA. And so they hook up the microRNA promoter to GFP and say, okay, here it is. And if we have a broadly expressed GFP under control of the target gene, and also express the Bantam microRNA, then we see repression in this zone. And now if we stop fussing around with reporter proteins and we actually go to look at expression of the ENA protein product, I think this is a protein, it might be an in situ hybridization, but if we look at expression in the normal situation, if you overexpress the Bantam microRNA, you reduce expression of the ENA product in this region. But the version of ENA that has that sequence in the 3' UTR mutated so that the Bantam can't bind it anymore, you don't see that stripe of inhibition. The discouraging thing was that you didn't see an increase of the ENA product throughout the embryo. So we know that from this that the microRNA can suppress expression, but we don't know that that's really functional. This is just going to go through a couple of papers that look at other aspects of using Cas9 in Drosophila. This one is looking at paired nicking. So two guide RNAs in close proximity to each other on opposite strands, and the D10A mutant of Cas9. And they find, as it was found in mammalian cells, that paired nicking does work, that you get heritable mutations for these are separations, the offset distance between the two guide RNAs. You do get mutagenesis by paired nicking. They characterize some of the products, but the efficiency is quite low compared to the individual guides with wild-type Cas9. So these are individual guides with wild-type Cas9, just looking at germline mutagenesis as a percent of all alleles, and you know, everything's at least in double figures, and some are up in the 80% range with wild-type Cas9 and the single guide RNAs, but the pairs never do better than about 20%, even in the range where the paired nicks are still treated as a double-strand break. So in this situation anyway, the paired nicking works. If you know exactly how to design it, it works fairly well, but you're losing a little bit of efficacy by going that direction. And in this case, they didn't test specificity, whether there was a gain in specificity. This paper talks a lot about specificity. So they are looking at a particular target. This is the guide RNA sequence, to see what's the effect of substitution mutations coming in from the 5' end of the guide. So these are different guide RNAs at the same target. And what they find is that one or two mismatches out at the 5' end decrease but don't make the mutagenesis go away. But as you begin making more mutations and moving them a little more into the body of the guide, that you're getting no mutagenesis at all. And these are at a number of different targets. The one or a couple of base pair mismatches way out at the 5' end sometimes has a very, very large effect on mutagenesis. And this is looking at on-target and off-target effects for a number of different genes. And so these were synthetic guide RNAs where they could place the mismatches where they wanted them. These are cases where you're looking at naturally existing genomic mismatches. And these situations are all ones, I think, where there are just 3 mismatches between the secondary target and the design target. And in all those cases, the mutation rate at the secondary target was undetectable. You have to know what the detection rate is. We're counting individual flies here, so if the detection, if the off-target effects were 1 in 1,000, they would not have been seen. Okay, but these guys also tested the truncated guide RNAs, and they find that the truncated guides often work just as well as the 20 nucleotide guides, and putting a mutation, a single 5' end mutation into the truncated guides seems to have a very large effect on mutagenesis, on efficacy. So again, sort of supporting the idea from the original truncated guide paper that making these truncations makes the system more sensitive to mismatches. So Barbara has looked at this for obvious reasons. What they're doing here is they're trying to assess what target sequences work best for Cas9 mutagenesis in Drosophila. And so they categorize things by the GC content of the first 6 base pairs upstream of the PAM, okay? And what they find is that if you only have 1 GC base pair, that the efficacy, I mean, lots of people wouldn't complain about this, but 50% mutagenesis, but they tend to be lower on average than targets where the GC content in those 6 base pairs is higher. And so this is a scatter plot of these observations. And what they say is that if you have not sure how they can get, I guess these aren't fractional, these must be 2/3. So if you have 4 or more GC pairs in those 6 base pairs, you're going to do measurably better than if you have fewer than 4. And then if you are Barbara or me, and you know what Barbara's group has done, you go looking to see does this correlate with GG in that 3' position next to the PAM. And there are some examples where those are amongst the good ones. Actually, this one doesn't happen to be all that good, but there are a lot of cases where very high levels from this group don't have GG in that position. Yeah. So let's see if we can find one. This has TC and is very good. TC is very good. CC is very good. So, they tend to think in their context that it's more than just the first 2. Maybe it's the first 6 you should be looking at. I haven't sat down. If I get fired and have nothing else to do, I'll sit down and I'll try to take everything I can find in the literature and see whether we can make correlations like this that are consistent within a species, a cell type, phase of the moon, some other variable that we might be able to correlate these things with. And this is a slide I like to show just to prove that my lab has also done the CRISPR system. So what we did was we said, okay, let's see if we can figure out which of these platforms does best. If there's one that always does best, we'll kick out the others. So we had these two sites in rosy and in yellow where we were doing quite well. So this is percent germline mutants, percent mutant alleles. And we were getting, in the experiments where we were doing direct comparisons, high frequencies of mutagenesis at rosy and pretty good at yellow. And we designed talons and guide RNAs to sit right on top of those sites as much as, as closely as we could, and then pick a couple of other sites that we didn't design ZFNs for, but we're comparing talons and CRISPRs. And what you can see is that at this site, the ZFNs won. Well, those are our best ever ZFNs, so not a fair comparison. But here the talons were doing better than Cas9 was. But when you go down to yellow, CRISPRs are the clear winners, and the talons, although they did quite well at 3 other sites in the genome, gave us no mutations there. And there's no obvious reason why those failed, although there are obviously some differences from site to site, and I can wave my hands at that and possibly explain it away. But the bottom line here is that you can't always predict the winner. And one thing people will frequently ask me about is whether everybody should just give up on talons and ZFNs and CRISPRs are going to dominate the field. Well, if you're setting up a lab and you want to do mutagenesis, the CRISPR platform is definitely the easiest of them to employ. But there are many situations where you might choose to use ZFNs or talons, and there are ZFNs and talons that are not going away soon. So. This is particularly true for situations where you have one target that you want to hit over and over again, and the development of the nuclease for that target is going to be a small portion of your program. In that situation, you don't care which platform you're using. You just want high efficacy and high specificity, whatever the platform is. Okay. There are web tools. These are some of them. And from these websites, you can get access to Cas9 being driven by various promoters, also the FOC1 fusion to Cas9 and the Cas9 nickase, different plasmids. You can use them for this Phi-C31 integration. That have Cas9 or the FOC1 Cas9 under different promoters. And so the Fly community is generally very generous in spreading technologies around. So as the tools are developed, they will be available. Paper appeared earlier this year in Science promoting this method for getting mutations at a particular locus in Drosophila rapidly spread through a population. And they call this the mutagenic chain reaction. And what it is, in other terms, is it's a gene drive. And I'm going to talk about that aspect of it in a moment. But let me describe it to you first. So you make a guide RNA that's going to make a break in a particular locus in the genome, and you deliver it along with Cas9 to the flies by embryo injection, presumably. But then you provide a donor DNA that has homology arms to this locus flanking the break, and between them, coding sequences for Cas9 and that same guide RNA. So the break is made, some frequency you'll get homologous repair, and now that locus is interrupted and also carries the functional Cas9 and guide RNA coding sequences. So if this happens in one allele in a fly, when you mate to another fly, the incoming allele of that same gene is now a target for these reagents. And it'll get cut. The previously mutated allele is now a template for repair, and you can get that homologous repair and the Cas9 and guide RNA now integrated at the other allele. So now you've got a fly that's homozygous for this, and it's going to transmit that process to all of its offspring, not just to half of them, like this fly did, and this will rapidly spread throughout the population because you've got the nuclease encoded within the mutation, okay? And if you're interrupting a coding sequence, now that interruption of the coding sequence is spread throughout the population, and this is a diagram of it. By normal Mendelian inheritance, you make a mutation, and it is transmitted, this is from a male transmitted into the female, but not the male progeny, and only linearly transmitted from here on. With this mutagenic chain reaction, the male gives the X chromosome to his female progeny, but they all get converted by this chain reaction. And so now what you expect, if that mutation was in the yellow gene, all the females will be phenotypically yellow, but the males who got their X chromosome from mom will be phenotypically wild type. And here are the flies. The females are all yellow, the males are all wild type. And there was one female escaper. And this is just the numerology of this. If you bring this MCR locus in from the female, now all of the offspring will be getting it. Okay, so that's cool. You can make a mutation and spread it rapidly into a population of flies and see what happens. But what if that fly came home on your shoulder and began to mate with the flies that are on your rotting bananas, or in my house it's usually the little bucket that's taking the apple peels and the bad spots out of the nectarines and things like that. So the fruit flies will come to fruit. So some of the wild type flies mate with one of these flies carrying this gene drive, and now we're going to get yellow mutants in the population. Well, what if that was some other kind of gene that was interrupted? Would that mutation is spread through the population? So a lot of concern has been focused on this because people are also thinking about gene drives as a way to control mosquito populations, particularly the mosquito vectors for tropical diseases. And so if you could identify a gene in a mosquito that was required, it wasn't essential for the mosquito's life, but was required for transmission of the malaria parasite, for example, you could put that mutation in one of these MCRs or gene drives into the population, and within a few generations, it would begin to spread through that mosquito population. But you got no way to stop the spread. Once it starts, it's on its own. So a very thoughtful young fellow he's a postdoc in George Church's lab, I think now may have his own group, named Kevin Esvelt, has been quite concerned about this and has written a few times. And he put together a paper with some other people. It's been submitted to Science. And in that, they proposed an alternative gene drive that would be more controllable. And the idea here is that you only put the guide RNA into the donor DNA, the coding sequence of the guide RNA, so that the guide sequence is being driven into the population, but it's only, the drive is only continuing as long as there are Cas9 expressors around. And so if you can control the presence of Cas9, you can stop the drive. And so in this paper, they're also recommending physical barriers, other type of possible genetic barriers, various ways of controlling the escape of flies that have this sort of thing going on. But it's something to think about, and it's something that has begun to get interest outside the scientific community. The general populace thinks we're creating creating monsters in the laboratory and want to convert all of them into monsters. That may be true for some of us, but mostly not. So, but when something like this comes along and scientists themselves call attention to it, it tends to get out into the public realm a little bit too. So I think Kevin was particularly interested in collecting members of the Drosophila community. There's a very long list of authors on this submission to say, look, it's smart to control this, and here's one element of that type of control. So that was sort of a long and winding road through how one can make use of Drosophila tools and our genome engineering tools to do things in this model organism. So. After the break, Randy Peterson will talk about the zebrafish, so we're moving up the phylogenetic scale a little slowly. And at the lunch break, we're going to offer the opportunity again for people who want to go on camera to talk a little bit about their experience in the workshop. So we're thinking of this, I mean, to be honest, we're thinking of this as collecting testimonials, you know, and we might put a few of your words up on the website, you know, with your talking head. But we'd just like to see if you're getting something out of the workshop and you'd like to help us spread the word around a little bit. You could just stand in front of the camera for a few minutes at the lunch break and tell us how you liked it. If you rant and rave at us, we'll film that as well. We may not post that. All right.